Welcome everyone to Iris Biotech's first online workshop this year. True to our slogan, empowering peptide innovation, we are keen on knowledge exchange and getting in contact with scientists all over the world, which is why we started this Iris Biotech workshop series already 2 years back. Today, our focus is on protein semisynthesis to study post-translational modifications, surrounded by some brief introduction information of our company, Iris Biotech. My name is Thomas Bruchdorfer, and I am the CSO of Iris Biotech. Today, I will guide you through this online workshop. If you wish to announce your current participation on this workshop here on social media, just like LinkedIn, for example, please feel free to use the hashtag Iris Workshop Series, which you should see right now in the Microsoft Teams chat. In order to have a pleasant experience for all participants, I would like to begin with some technical guidelines. For the best possible audio and video quality, all participants are automatically muted and your video camera is switched off. Another important information is that this live event is recorded. Therefore, if you enter late or miss anything, please check on the bottom left side of your Teams screen if you're watching the recording or if you are live. You have the possibility to pause the video, to rewind or jump back to the live session. In addition, the recording of the workshop will be uploaded on our homepage just afterwards. Questions can always be submitted by using the Q&A chat of Microsoft Teams. You can either enter your name or submit your questions anonymously, just as you prefer. We will frequently check these questions, publish them, and submit them to the respective speaker. If you have any technical queries, you can also submit them in this Q&A chat. Our technical assistants will then get in contact with you and try to resolve this problem. Initially, the questions are just visible for the organizers, and we will then publish them to the whole audience. So don't worry if your question just takes a few moments to appear. To try out this Q&A chat, we will now post a question which you can answer. So you should see now the question. The question is, who is today's external guest speaker within the chat? Please type your answer now in the Q&A chat. All right, we see already some answers coming in, and of course the right answer is Christian Becker. So as this seems to work, I would like to quickly give you an overview on today's agenda. We will start with a short company and topic introduction by our head of marketing and sales, Karin Rustler. Afterwards, our external speaker, Professor Christian Becker, will provide detailed information on his research at the Institute of Biological Chemistry at the University of Vienna, Austria. As mentioned, questions will be collected through our talks in the Q&A chat and are forwarded to the speakers. Of course, you can also get in contact with us anytime later via email or phone call. And one last note from my side. If you accidentally close your Microsoft Teams, don't worry, you always can re-log in. And as this workshop is also available as recording, you will not miss anything. So after this quick technical introduction, let's continue with the first agenda point, an introduction to our company by our head of marketing and sales, Dr. Karin Rustler. Karin studied biochemistry at the University of Regensburg in Germany and obtained her doctoral degree in organic chemistry for the synthesis and characterization of photoswitchable compounds. She joined Iris Biotech in 2020 and is today our head of marketing and sales. So Karin, please share your presentation and please go ahead.
So thank you, Thomas, for the kind introduction, and also welcome from my side to today's online workshop. Within the next slides, I want to give you a brief introduction to the company Iris Biotech, as well as a very short introduction to post-translational modifications. Iris Biotech has been founded in 2001. In 2013, due to the growing demand for customized products, we started the Iris Biotech laboratories, which are located in Willstedt, close to the French border, whereas Iris Biotech is located in Magdretwitz, so close to the Czech border. Our portfolio comprises more than 6,000 products. To realize this, we are working together with 15 manufacturing partners around the globe, which we select very carefully. Having only qualified partners on board allows us to realize a complaint rate consistently below 0.5%. In 2021, we refreshed our company logo and switched to a more modern design. So on the top left side, you can still see the old one compared to the new and more modern one on the bottom right side. To give you not only facts and figures, I also want to show you the beautiful landscapes we are surrounded by, as well as a picture of our company building in Magdretwitz. We see ourselves as qualified supplier and competent consultant and want to support our customers in the design and execution of their projects, no matter if it's about small research scale or commercial bulk production. Our portfolio provides building blocks and reagents for peptide synthesis and modification, but also advanced linkers for conjugation, as well as drug delivery technologies, all rounded up by our custom synthesis service. Besides, we are providing our self-written brochures and flyers, free for download on our website to promote knowledge exchange, true to our slogan, empowering peptide innovation. Collaborating with research groups all over the world and acting as platform for their innovations allows us to make latest technologies and building blocks available for the whole peptide community. One of the groups we are collaborating with is the research group of Professor Christian Becker, today's external guest speaker, talking about protein semisynthesis to study post-translational modifications. And also exactly these workshops are an important feature for knowledge exchange. Before I pass on to Christian, I would like to give you a brief introduction to post-translational modification. Synthesized peptides can be further processed by post-translational modification, which leads to a high diversity of existing peptides. Post-translational modification includes the covalent addition of functional groups or the proteolytic cleavage of the premature polypeptide chain, for example. All these modifications are essential for the activity regulation and function of a protein. Typically, post-translational modifications are enzyme-mediated, but there are also non-enzymatic post-translational modifications, which occur, for example, through aging, diseases such as diabetes or cancer, or exposure to heat. These are often unwanted reactions with metabolic compounds. One example is the reaction of, for example, surface-exposed lysine or arginine side chains with the metabolite methylglyoxal, leading to the formation of argpyrimidine. To investigate biological effects of, for example, the argpyrimidine formation, it is really important to have such building blocks commercially available for synthesis. So I would like to end my introduction showing you one argpyrimidine derivative, which is provided by the research group of Professor Becker and commercially available at Iris Biotech. And with this, I would like to thank you for your kind attention. And yeah, Christian is the expert for this topic. So we are very much looking forward to hearing about his research. Thank you.
All right, great. Thank you, Karin, for giving us this introduction. And as I see so far, there are no questions. Let's go on with today's highlight and switch to the external presentation. It is a real pleasure to me to introduce Professor Christian Becker, head of the Institute of Biological Chemistry at the University of Vienna in Austria and of Vienna Doctoral School in Chemistry. Christian studied chemistry at the University of Dortmund in Germany, obtained diploma in 1998 and PhD in 2001. He then became a postdoctoral fellow with Griffin Therapeutics until 2003, and then started his independent career as a group leader at the Max Planck Institute in Dortmund. In 2007, he was appointed as professor for protein chemistry at the Technical University Munich. And in 2011, he became professor and head of the Institute of Biological Chemistry at the University of Vienna. And in 2020, he became then the founding head of the Vienna Doctoral School in Chemistry. His group develops and uses chemicals as well as biochemical means to generate peptides and proteins with otherwise unattainable post-translational modifications to address biomedical and biotechnological challenges. Christian, it's a true pleasure to host you as our guest speaker today. So can you please share your presentation with us?
Yes, of course. Thank you very much.
Okay, yeah, this seems to work. Thank you. So for the audience, if you have any questions, please post them in the Q&A chat. We will then later answer them. Christian, the floor is yours. Please go ahead.
Thank you very much. Thank you all for giving me the opportunity to present our research in this biotech workshop series that IRIS has set up. We already heard a very nice introduction about the different varieties of post-translational modifications and why we need protein chemistry to study their function and also their physiological roles. And I will take some time today to introduce you to these topics and also provide a little bit of background on protein chemistry. And of course, we'll also present two projects in which we deal with introducing post-translational modifications into specific proteins, and hopefully can tell you also a little bit about the challenges along the way, a little less about the biochemistry and physiology that results from this, because I would like to focus more on the, let's say, chemical and synthesis challenges related to these projects. But first of all, just a quick overview on the work we do in my group at the University of Vienna. It's roughly divided into three big areas, and the ones on the left and on the right play no role in today's presentation. I will solely focus on the protein chemistry part that you see in the middle here, and therefore just jump over the slide now and go onto a slide where we see an historic representation of the development of peptide chemistry, which is somehow biased based on the input of the people who have actually created this, Steve Kent and Phil Dawson, some of the major players in protein synthesis over the past 30 years. And as you can see here, it all starts out with Emil Fischer's work on peptides, and then slowly develops via introducing the urethane-based protecting groups into peptide chemistry. Then, of course, the key finding of Bruce Merrifield about solid phase peptide synthesis that had only changed peptide synthesis, but many other areas of chemistry. All of them, of course, related still to the fact that we could produce a quite large number of peptides based on solid phase synthesis, but still not very long peptides, or at least not sufficiently long to cover functional domains of proteins or even full-length proteins. And this you see in this graph as well. The realm of functional domains starts about 110 kilodaltons, so roughly at 100 amino acids. And the average protein size in humans is about 30 kilodaltons, so roughly 300 plus amino acids. All of this is still very difficult to obtain by solid phase peptide synthesis. The landscape of generating such large polypeptide chains synthetically has changed quite dramatically in the early 1990s by introducing so-called chemoselective ligation strategies. And maybe I switch already to the next part of this slide. Because before that, all people involved in peptide science typically envisioned to link fully protected peptide segments to obtain larger proteins, which comes with a challenge of solubility in handling these protected peptides, which is only possible to up to a certain length, and then becomes unfeasible. And back in the time, the idea to use fully deprotected and thereby mostly water-soluble peptide segments, but introduce reactivities into these peptides that would allow you to link them together via their unique reactivities came up and was further developed over the past 30 years quite dramatically. And in this schematic picture on the right here, you see what is required for that: fully deprotected in aqueous buffers, and then bring together. So only the red and blue functional groups react with each other, and these reactions can then lead to a large variety of different functional or to a different chemical moieties that you see here. So you can go through oxo or thiazolidines, you can go to thioethers, thioesters, triazoles, and so on. But of course, if you would like a native amide bond at that position, this is the most preferred solution for these kind of linkages. This solution was offered in the early 1990s by introducing native chemical ligation that many of you have probably heard about, and that thousands of times to make proteins by either chemical synthesis or just very briefly after introducing this chemistry for synthetic peptides, also extended to actually being able to combine recombinantly produced peptide segments with synthetic peptides, which again allows to access much larger protein segments by recombinant expression and only concentrate on the synthetic parts wherever you would probably do some changes to either side chains or the backbone and so on. So to site-specifically introduce modifications. And this is essentially what we now consider protein semisynthesis, where we link synthetic peptides to recombinantly produced proteins in different ways. And I'll briefly go into that in a second as well. The different ways are essentially dependent on how you generate the synthetic or recombinant parts. And this slide is quite busy, but it summarizes that you can do this for all kinds of segments. In this case, you could link a recombinantly produced protein of interest, the POI1 here, that is initially expressed in fusion with an intein, and the intein is the functional moiety that allows what is called expressed protein ligation, because it gives you a tool to generate recombinant protein thioesters, as you see here. Without going too much into the underlying mechanism, these inteins can cleave peptide bonds via thioester intermediate, and that intermediate can be cleaved by addition of a high concentration of small molecule thiols, as shown here. And then from this, you derive a protein with a C-terminal thioester, which is the unique functionality that leads to native amide bonds, as you see here. Via reaction, as it is drawn here, you link this to a second peptide with an N-terminal cysteine that undergoes a thioester exchange and then an acyl transfer from the sulfur to the amine, and then you generate an amide bond. And this is done here for an N-terminal modification of this synthetic peptide shown in red. But of course, this synthetic peptide, if appropriately protected, as shown here, can also be modified on the C-terminus previous to the N-terminal modification. And I should mention here that the order of ligation reactions you can actually change however you prefer it. And here we see another recombinantly produced segment, our protein of interest, part 3, where you need to install or release an N-terminal cysteine as well. There are different ways of doing this. And this then undergoes another native chemical ligation reaction, forming an amide bond. And in the end, you have now introduced a synthetic segment that allows you full freedom to modify the peptide chain with any kind of building block that is available to you, or with the whole toolbox of organic chemistry, and to introduce it in the middle of two recombinant segments. And of course, you could now also envision that there might also be an N-terminal extension on this side, and so on. Of course, this is probably the most complicated case, introducing a synthetic peptide in the middle of two recombinant segments. And later on, I will deal with two examples where we install a synthetic peptide on the N-terminus and one on the C-terminus, with each of them carrying different post-translational modifications to also the title of the talk. This of course, comes along with the fact that if you want to do that, you need to be able to handle both parts, or both worlds, I should say, that are required here. On the one hand side, you need to be able to actually manipulate on a genetic level your cells, your expression systems, and then you need to be able to recombinantly express proteins in sufficient amounts. And these are typically high milligram amounts to actually gain access to materials that then can later be transferred to biochemical assays and even structured analysis and so on. So you need to be able to do also molecular biology, protein purification, and so on for the recombinant segments. And on the other hand, of course, you need to be able to do the chemical synthesis, and that does not only start with making peptides, but it also starts with generating certain building blocks for peptide synthesis. And if you can do that, in the end, you're able to address a variety of post-translational modifications and proteins. And here are a couple of examples from our group. And today I will discuss two examples. One is shown on the lower right. This is GCSF. And one is this protein over here on the left, which is HSP27. And I will say more about these proteins in a few minutes. Post-translational modifications were already introduced very nicely today, and this is just a summary of, I would rather say, a random selection of modifications you can introduce, and you see glycans, lipids, phosphorylations, variants of phosphorylations. You even see ubiquitination here, so attachment of a whole protein. The enzyme-mediated modifications of proteins, which we heard about. And this is also our first example that we will deal with. And you can already tell from the structure of these glycans here that these modifications can be quite complex. And this is, together with being able to install them site selectively in a large protein, another challenge that comes along with this. And here I just summarize what I just said. So the basic concept in protein semisynthesis is essentially to produce suitable protein segments if they have no modifications and if they are rather long. The way to use here, at least based on our experience, is recombinant expression, where we are able to generate the chemical moieties for ligation to synthetic segments, either by INTI infusion or by protease cleavage to release N-terminal cysteines or C-terminal thioesters, respectively. And I will show you later on that there are also other options where you don't use necessarily thioesters, but you can also use other functionalities. And the other part you need is a suitably modified synthetic peptide segment, for example, this glycosylated synthetic. You would like to stitch these together to give the full-length protein sequence with your desired modifications. In this case, we see a glycosylation here and maybe a fluorescent probe down here with this orange, or indicated in orange here. And an important fact that I have not mentioned before is that generating these segments, linking them together, can already be a challenge and is typically a unique process for every single protein because they behave differently depending on their sequence. But Then there is a second step after being able to synthesize the whole sequence. You need to be able to fold the proteins, because if you can make the polypeptide chain, it doesn't mean you can make a biologically functional molecule, because this requires folding of the protein, at least in most cases. And this is something that needs to be done after successful synthesis, purification, and so on. And is also unique for every protein, comes with different challenges. Fortunately, so far in our hands, we never had a protein that we were unable to fold, but I should say that the range of folding yields can be extremely variable. So it can be you dissolve the polypeptide chain in an aqueous buffer with controlled pH, and the protein folds. No loss of protein, 100% conversion into the species you would like to analyze further. But it can also be that 80 or 90% of your protein drop out of solution, even if you don't do a direct dissolution, but go via different dilution steps or even dialysis steps, and all the kinds of tricks that people have developed for protein folding. So the good news is all of proteins we have made so far can be folded. The not so good news is that folding yields can vary quite a bit. And at that stage, I should also mention, of course, that these are not the only methods to introduce modifications into polypeptide chains. There is a large area that quickly developed. I would refer to as genetic code expansion, that I will not discuss today, but that also allows you the site-specific incorporation of modifications to proteins via expression. So there is almost no synthesis involved, maybe besides the building blocks that you need to generate somehow, the amino acid building block. But these systems are also challenging because you need to develop the tools to actually incorporate new and modified amino acids for every single protein, again, to get these modifications installed. And of course, it comes with challenges in yields and so on as well. But this is a quickly developing field that I need to mention here. Another approach is much older than semisynthesis or protein synthesis, but also older than the genetic code expansion that I just briefly described. It's protein modification by relying on the available functional groups in expressed proteins. And probably the most commonly used one is a thiol group of cysteine, and here are a couple of examples. And there's chemistry to address thiol groups selectively, chemoselectively, I should say, for more than 100 years. And this chemistry is still evolving and allows you to introduce modifications as well. Most likely not native modifications, even though we have developed methods to do so, and others, of course, as well, to introduce, for example, granulations into proteins by using cysteine-specific chemistry. But this is something I will not go further into today. The remaining two panels here just summarize ligation chemistry again. On the lower left, native chemical ligation. On the lower right, another example for such a 3-segment ligation with 2 recombinant parts that I described earlier. So I don't spend more time on this, but rather jump into our first example. And this is this granulocyte colony stimulating factor, in short GCSF. It's a cytokine with a multitude of physiological functions. The best known one is probably the one why it's also therapeutically used, and it's used to treat neutropenia. So it actually stimulates the growth of immune cells, white blood cells. And it's has a huge market value. It's what we call a blockbuster drug. So it has $1 billion market or a multi-billion-dollar market share. And mainly used to treat people also under chemotherapy and so on, to stimulate the growth of immune cells. GCSF is also a quite challenging protein synthesis target, and it carries post-translational modifications, namely O-glycosylations. And there are, let's say, 3 variants on the market, actually approved, and you can go to the pharmacy and get these if you have a prescription for them. These are made by recombinant expression in either E. coli or in eukaryotic cells. The one in eukaryotic cells carries a certain pattern, not fully homogeneous, but in a, let's say, well-defined homogeneity window, O-glycan structures. And then there's also a product that is based on the E. coli protein that gets pegylated. But in the end, even with these products in hand, nobody has yet fully understood how glycosylation affects protein function in this case, and if that would interfere, for example, with receptor binding, or how it stabilizes protein folding and so on. And this is why we thought this would be a good example to bring together chemistry that we have developed over the past years. Namely chemistry to introduce such O-glycosylations in a chemoenzymatic fashion, and on the other hand, of course, to use our protein synthesis expertise to link the protein together by using a large recombinant part and a small synthetic part. I should also mention that there has been efforts to do this fully synthetically, because the protein is not that big. It's 174 amino acids, as you see here. There's one published synthesis by the Danishevsky group, which has reported a lot of challenges along the way with handling of the different synthetic peptides due to the large number of cysteines in the sequence and so on. And maybe this is a time to look at the sequence of GCSF, which you see now in the top left. And here you see all the cysteines in the sequence, which we typically like because cysteines are always potential ligation sites. But in this case, the cysteines are not very well for ligations because they are all towards the N-terminal part. And unfortunately, the glycosylation sits on this threonine 133 here, shown in red. And therefore, we initially selected this glycine site here as a ligation site for our initial synthesis route. The other colored amino acids I will mention later. And what we wanted to achieve was to introduce the typically found O-glycans onto this threonine side chain. So we find there the TN antigen, also the sialylated TN antigen. And these we would produce chemoenzymatically by extending this building block that carries the initial carbohydrate, and then we would use enzymes to attach the next one. I will talk about this in a second a little more. And this was the initial synthesis strategy. As I told you, the N-terminal part with all the cysteines recombinantly expressed, so to avoid all the challenges during the synthesis with handling and so on. This works. It also doesn't give great yields. Great yields would be, I don't know, 20, 30, 40 milligrams of the protein thioester from a liter of expression in E. coli. This we do not get here. We get a couple of milligrams out of this, but it's sufficient to carry the synthesis on. And on the other side, we would make a synthetic segment, including our site for glycosylation. And as we have a glycine here on the ligation site, which typically is not suitable for native chemical ligation, we need to introduce such an auxiliary that would allow us to release the glycine after synthesis, but also give us the opportunity to use this thiol moiety here for the initial trans esterification step that I've shown you briefly before. And also a pegylation, which we will use for chemoenzymatic modification. I show you in a second how that's supposed to work. Unfortunately, this was a strategy that was supposed to work out well, but did not, because the N-terminal segment has ability, so we could not really bring it up to the millimolar concentrations that we typically like to have for these kind of ligation reactions. It's a bimolecular reaction, so high reactant concentration really drive it quickly. Therefore, it took a long time and gave only a small amount of ligation product. So we lose a lot of the valuable segments and C-terminal segments. And also there were additional challenges with releasing the auxiliary in the presence of all the cysteines. So in the end, we could not obtain the full-length protein in any sufficient amounts from this synthesis and had to reevaluate how to do this. Before I tell you how we actually succeeded with this, just a quick look at our auxiliary that we used for this. This is a molecule and it links to the peptide chain via this amino group here, and I'll show you that. So this is it's the amino group to link it to the peptide, and then we have this PEG chain here that we actually need for efficient chemoenzymatic glycosylation. And this chemoenzymatic glycosylation we do in solution, so homogeneous catalysis. We add the activated sugar, we add the glycosyltransferase, the suitable one, and fortunately nowadays there's a large number of them available for extension of glycans on peptides and proteins. And if we attach one of them, we can just precipitate all the contents of the solution by adding ether or a mixture of ether and ethanol, and then resolubilize, and only our peptide then carrying the additional glycan or the additional carbohydrate on the glycan gets resolubilized, and you can add the next glycosyltransferase and activated sugar, and thereby extend your carbohydrate chain quantitatively and very efficiently without any additional purification steps, chromatographic steps, and so on. It's just a precipitation and dissolution or resolubilization approach. This is why we need the PEG. It needs to be there for this resolubilization, otherwise this doesn't work. In the next step, of course, we would then need our thiol group here for transesterification, and then the acyl transfer to the amino group. And this works with this auxiliary, but is not as efficient as the normal cysteine in this reaction, because we have a secondary amine here sterically hindered. So this acyl transfer step that we need to form this amide bond here is typically much slower, and this was also part of the reason why our initial strategy didn't work out well. Sorry. So the end, if everything works well, we illuminate, and this is why we need this nitrobenzyl scaffold here to release all of this auxiliary together with the PEG chain and obtain the native polypeptide with our carbohydrate moieties. By just telling you now that our initial strategy didn't work, we had to come up with an alternative that would allow us to introduce a PEG chain, but also would allow us presence of all the cysteines. And one alternative that has been explored by several other groups, and I show here pictures from publications of the Dawson group, the Metanis group, the Payne group, and so on, that have shown the value of selenocysteine in native chemical ligation reactions, where the selenol takes over the role of the sulfur or thiol group in attacking the thioester, forming a selenoester intermediate and then an acyl transfer. And the really valuable part here is that the selenol can be easily deselenized in the presence of other cysteines without touching them. So in the end, you can remove or convert the selenocysteine into an alanine. And this is what we have done. We have slightly shifted our ligation site, so we could now use a native alanine for this kind of chemistry. And this is this busy slide here. In the beginning, same as I've shown you before, the GCSF is released from an inti fusion construct. You see a slight change in numbers because we have to move the ligation site 2 amino acids up to hit an alanine, and we generate the thioester, and then we make synthetically a peptide with our glycosylation at position threonine 133 and an N-terminal selenocysteine. The selenocysteine comes with some challenges regarding the protecting group. If you remove the protecting group, it forms a dimer very quickly, a diselenide, that needs to be reduced to be active in the ligation chemistry. But this works, and we were actually able to obtain our first full-length GCSF in reasonable amounts, including the first carbohydrate moiety here. But of course, for chemoenzymatic extension of that carbohydrate to generate all the different versions we wanted to obtain, we would need to add along the glycosyltransferase and so on, and we need to be able to actually carry it through this precipitation and resolubilization process, and therefore we need the TEF. And in order to do so, we just went back to a strategy that we used many years ago before introducing that photocleavable auxiliary. Just added the PEG chain to the N-terminus via a TEF protease cleavage site so that this could be removed by proteolysis after we have extended our glycan chain. We did this, glycosylation worked well, but unfortunately the TEF cleavage didn't work out here. So the selenocysteine could not be released. And so far we have not been really able to release an N-terminal selenocysteine from a TEF site in such a construct. Fortunately, at the same time, another PhD student in the group developed such a building block based on the auxiliary here, or similar to the auxiliary I've shown you before. Again, a nitrobenzyl scaffold that can be attached to a side chain. In this case, to a glutamic acid that then converts to a glutamine. So we used a native glutamine to introduce this side chain linker together with our PEG chain. And with this, we now had all the necessary features in our synthetic GCSF segments, the N-terminal selenocysteine for ligation and the PEG chain for glycosylation. Did the glycosylation, then had to do the ligation with our segment here, and then could do the desalinization and really obtain the glycosylated GCSF variants. And just as a short sidestep, these kind of sidechain linkers to introduce tags like the PEG chain here are not new. Many people have used them before to solubilize hydrophobic peptide segments, and there's a large variety, and I will not discuss them here. Just to give you a brief idea on how this linker synthesis works out and what kind of peptides we make out of this. So this is the quality of peptides that we start with. And it's really essential to have high quality synthetic peptides as well as high quality recombinant protein segments to not really make the ligation and the subsequent purification more challenging than necessary. So start with the highest purity that you can get your hands on if possible. Just to show you the assembly reaction, here you see the recombinant thioester. This is our synthetic glycosylated segment. It works. It still takes that we have used before. That's already a little better. And it gives nice conversion to the desired final product. We're able to make different versions of GCSF by doing so. And in the interest of time today, I'm not going into too much detail on the analysis. It should be suffice to say that they all folded like the wild type, that they behaved very similar, that the glycans, at least in vitro, showed an effect where we see less effect, can show them to be less effective than the non-glycosylated versions, but this is only data based on cell proliferation assays in vitro. And at this, I stop with interpretation of the biochemical data. Happy to discuss this if you have questions about this. So in the end, we can now modulate GCSF solubility, at least of the C-terminal peptide via pegylation. We can introduce glycans chemoenzymatically. We can release our linker by photocleavage. We can use selenocysteine for ligation and so on. But at that stage, we're still not happy because the ligation was kind of slow. And fortunately, at the same time, together with the group of Richard Payne in Sydney, we were looking into the opportunity to convert recombinant proteins also into selenoesters, because Richard Payne has some time ago, together with the Metanis group, introduced ligations where they not only use N-terminal selenocysteine, but also C-terminal selenoesters. So these diselenide selenoester ligation is typically much faster than native chemical ligation based on thioesters and thiols. This is to do with the properties of the selenium, redox potential, nucleophilicity, and so on, and allows these reactions to run at much lower concentration, but could be really an advantage for the GCSF ligation, but also for many other protein semisyntheses. And therefore, we were interested in generating selenoesters on protein, on recombinant proteins. And there's some precedents on this, and the reaction that really helps the most is the one that you see here on the bottom, recently introduced by the Dawson group and also already used by the Chin group to introduce selenoesters into synthetic peptides. Unfortunately, we were able to show that you can actually do that also with using this Knorr-pyrazole chemistry into recombinant proteins. And here you see on the left how that's supposed to work. You see a gel of this kind of conversion to the hydrazide, and then you see the hydrazide to the ubiquitin ester transformation, and that works very nicely, very clean, and we have done that with very different proteins, and gives in all cases the desired seleno esters in nice yields. Also for GCSF, and just a quick glimpse on this, so we actually generated here the synthetic part with, in this case, a different glycan, a sialyl TN antigen, and then did a ligation with the seleno ester peptide, and this works at much lower concentration, much higher speed to give the desired product. And this is just a crude product, and we termed this the express protein seleno ester ligation, which we can hopefully now extend on also for more challenging protein targets. And this is the story about the EPSL reaction, so lower concentrations. In the future, you can imagine that with beta-seleno amino acids and seleno-containing auxiliaries, you could also further increase flexibility of these reactions to address even more challenging protein targets. And I know I've already extended my time quite a bit, so I will go through the second part of my presentation quite quickly. Because this is another example of a post-translational modification, but in this case, really one that addresses non-enzymatic PTMs. And we have heard in the very nice introduction already that these typically result from reaction with carbonyl-containing metabolites, as they are shown here. And they typically happen to proteins or are only found in proteins that are sufficiently long-lived and not degraded quickly, because then we have no time to find these modified versions. But if you have proteins that hang around for quite some time, or even for all of our lives, if we look at humans, these can be accumulated and of course also found during analysis, and has been analyzed by physicians and people who are interested in food chemistry, physiology, and so on for decades already, but has not been much in protein chemistry, and therefore we don't have that much data on the physiological relevance of these modifications. But you find them in all long-lived proteins that can be proteins that aggregate in neurodegeneration, so in tau, alpha-synuclein, and so on, but it can also be in collagen or in the proteins in our eye lenses and so on. And therefore, we believe that this is definitely something to look into and see how relevant are these different modifications. And they are complex. There's a large variety of them, so you have to start somewhere. And we have started to look into, for example, these carboxymethylations. We also started to look into this pyrimidine formation on the group of the arginine side chain. And in the end, there's also a large different class of cross-linking modifications that are shown here and that are much more challenging to look into. Most of what is on this slide I already said. So these modifications are either linked to protein aging, so aging of the organism itself, but less flexibility of collagen fibers, developing cataract due to modifications of eye lens proteins, and so on. All of this is related to aging. But also in disease states, you find elevated levels of such modifications, not surprisingly, because typically metabolism is also much faster in, for example, cancer cells. So you also have more metabolites that can potentially react with your proteins. To investigate these in a site-specific manner, you need suitable tools for that, the building blocks, and you need to be able to put them into your proteins at specific sites. And this is something that we have done over the past years. And before that, there have been studies on these kind of modifications by simply taking a protein, incubating it with an electrophilic reagent, in some cases even heating it up, and then you come up with a complex mixture of modified proteins. So you have no homogeneity in this. You cannot really pinpoint functional consequences to certain modifications. The only thing you learn from this, if you choose the right conditions, you learn exposed and potentially sensitive to such modifications, which could also be a relevant information, because even appear to be random, these modifications, of course, depend on accessibility of certain side chains, of how pKa values are shifted in folded proteins and so on. So it's really not a completely random modification, but something that really depends on the protein structure. But to really introduce this at a single site to then analyze such a single site modification, you would need a new building block. And this is what we started out with many years ago. Postdoc in the lab started with the synthesis, and we have already seen the building block initially that is also available via IRIS. And this we use now also to study the small heat shock protein 27, which is involved in many processes trying to save proteins from misfolding. It's also linked to a couple of disease states, which I cannot go into detail here. Here you see the sequence, and we have introduced together with colleagues a large variety of different modifications that I'll just summarize here. And I will not discuss most of them. I will just focus on this arc pyrimidine modification that I've just shown you, and especially on the one in the C-terminal part here. We also now analyzed some in the N-terminal part and introduced it in the middle and so on, but this is something that I will not talk about today. How do you do that? Again, you take, in this case, a protein with a INTI infusion construct, generate the thioester, introduce a synthetic segment by, in this case, FMOC solid phase peptide synthesis, do a ligation, classic native chemical ligation, desulfurization to remove the non-native cysteine residues that we have used here for ligation. At the same time, you convert a native cysteine into an alanine, and I will come back to that later. And this is the building block I've shown. This is the data on the peptides. Again, high quality peptides really help to facilitate these reactions. And you do the ligation reactions. And here is the final product that you obtain. High purity again. The only inhomogeneity you see here is based on processing of the recombinant segment, because in E. coli, sometimes the N-terminal methionine residue that is linked to the start of protein expression is cleaved off, and this is what you see here. And to a small extent, it's still left on, but this, as far as we can see, has no functional consequences for the protein. And I mentioned already that during the desulfurization process that we use to get rid of our non-native cysteine residue here, we also convert an internal cysteine, the only one in the whole protein, into an alanine. And this is something where there's physiological uncertainty how relevant this is for function of the protein. A large part of the literature says, don't worry about the cysteine, you can mutate it, it has no impact on function. Others say it might have impact when there is initial dimer formation, because this HSP27 forms large oligomers, and it's important for initial dimerization and then oligomer formation. Therefore, we thought, let's make a version where this cysteine is also maintained, and therefore we introduced a strategy where you can, in the recombinant segment, introduce a protecting group, carry this through the native chemical ligation and desulfurization procedure, and then only release the cysteine later on with a selective deprotection, and thereby introduce the arc pyrimidine in this position where there is a body of evidence that this is also found in tissue, and then release a protecting group and have really the native protein available. We have done that by using this PAC protecting group. We also explored the trityl protecting group because both have been reported to be suitable for these kind of things. Trityl has not survived our desulfurization conditions, but the PAC group we have now used in several recombinant proteins, and that works very well. It's easily introduced. There's sometimes a challenge with the removal because it's a heterogeneous reaction with zinc as a catalyst, but if you get the conditions right, this also works nicely. Then you fold the protein, as you can see here, you ability, and we see that the API modification, the single one in one position, has not much impact on folding or on the thermal stability. But in a aggregation assay where we take a look at chaperone activity of our HSP27 on a substrate or client protein citrate synthase, we actually see quite an effect. The activity decreases considerably compared to wild type. So this is the wild type protein in black, and this is the aggregation kinetics in presence of the API modified protein. So it's a worse chaperone than our wild type protein. So obviously this really leads to certain loss of chaperone activity and is therefore probably not helpful for the cell if this accumulates in large amounts in the HSP27. We have looked into the background of why this happens. Obviously, there is a change in oligomerization of the API modified protein. And we also did some MD simulations and so on, because high resolution structures of this oligomeric protein is difficult to obtain. There are some recent analog structures that have come out by electron microscopy, but it doesn't form really fully homogeneous oligomers, but they can span a certain range of oligomeric states, which makes analysis quite challenging. This is one part. I also told you we have now looked at the N-terminal part, and there we have 5 arginine residues, and we have looked at all of them in terms of activity, and therefore have used an N-terminal synthetic segment that we converted to thioester. And in this segment, we can introduce API modifications at will in any of these positions or in all of them, and then link them to a C-terminally produced or recombinant produced C-terminal segment that we where we generate an N-terminal cysteine via a TEF site, and then again desulfurization and so on. And this works nicely. Unpublished data just at the very end, we see that these N-terminal modifications actually change our chaperone into a much better chaperone. So there are different modes of action here that are involved with this. So in the end, we generated with modifications in all, arc-pyrimidine modifications in all of these arginine positions, something like a super chaperone, which obviously binds certain client proteins, helps them to prevent aggregation much more efficiently than the wild type protein. And this is where I will leave this for today, just summarizing, right? So we can introduce this API modification based on the building blocks that we have derived site-specifically into a variety of proteins using protein semisynthesis, and now start exploring the biochemical and physiological background of these modifications. With this, I'll just need to say thank you to all the people who have done the hard work. So Maria has started the building block synthesis and prepared the first API modified HSP27. Somnat has taken over and done all the N-terminal modifications. Claudia has introduced the auxiliary and developed it further. Lucas has then done the GCSF synthesis with the help of Max. Susanne has helped with the selenoester generation, and Gerhard really has been involved in all of the expression project and so on. And here are important collaborations. Richard Payne I mentioned, Matthew Pratt we collaborated on with the HSP27, and same with Johannes Buchner. And of course, all these agencies and institutions for their financial support. Last but not least, I would like to finish up with a slide in my own interest. You heard in the beginning that I'm heading the doctoral school in chemistry at our department. And there's just now a call open. There are 21 PhD positions in all areas of chemistry open, so also peptide, protein chemistry, but also organic synthesis, materials chemistry, and so on. So if any one of you who's listening in today is interested, please check it out. It's open until April 7th, and we are happy to see your application. And with this, I'm at the end of my presentation and happy to answer questions.
Yeah, great, Christian. This was really a super overview, and I really admire your interdisciplinary attitude in your group. You're combining biological production and chemical production methodologies, and by this you're really pushing the boundaries of making peptides to, let's say, in a chemical way you can make some 30, 40, eventually 50 MERS, but for you it seems to be a piece of cake to make 100 or 200 MERS with just any modifications here and there.
Thank you, Thomas. I would disagree that it's a piece of cake, but maybe we can discuss this in more detail. Thank you.
And I see a couple of questions. Let me summarize them a little bit. So some of them are related to making selenoesters, of recombinant proteins. So if you're generating the selenoesters of recombinant proteins, do you see any undesired side reactions, or what are the typical hurdles which you need to overcome if you make selenoesters of recombinant proteins?
I go back to maybe the slides that shows a little more information on that. Sorry for taking some time here. But maybe this is the one that we should use here. So to address the first part of the question directly, we don't see any problems or undesired modifications in step here. So what I have not mentioned in between is that we do not see conversion via any kind of thiol or thioester intermediate, but what we do here, different to what I've shown you before for the sulfur-based ligations, is we cleave the protein into infusion construct with hydrazine as a nucleophile, not with a thiol. And that works very well. It also gives less hydrolysis and so on. So the resulting hydrazide then is converted to the selenoester via this knorr-pyrazole chemistry. And that works very well in our hands. So in this reaction, we don't see any problems so far, no undesired side reactions and so on. And even this selenoester is quite stable as long as you don't go to any extreme pH ranges and so on. It can also be stored and then subsequently used for ligations. Where we actually sometimes have to be careful is when we do the hydrazine treatment with our protein into infusion constructs for the cleavage, because if you don't get the pH right, which should be somewhere around pH 9 to 10, not much higher than that, and only short duration, because it's a very efficient cleavage typically. If you incubate it for too long, have a too high hydrazine concentration, pH too high, and so on, then we sometimes see the attachment or formation of additional hydrazide. We suspect that this is a reaction that happens on amino acid side chains, aspartate, glutamate, and so on, and you get hydrozides there. And of course, if you then carry them through to the next reaction, they can also convert into selenoesters, and then suddenly you have a protein segment with not a C-terminal selenoester, but with additional selenoesters on the side chains. And this is, of course, undesired. And therefore, you have to get this cleavage reaction right, which we have been able to do by carefully adjusting reaction conditions. Yep.
Mm-hmm. I think another key in this synthesis is the formation of the pyrazole out of the hydrazide. Which is, I think, going quite smoothly. And another alternative is to make the acid oxidation with nitrate, which probably is causing more oxidation side reactions on some other parts in the peptide.
Yeah. I guess I mean, of course, the pain group has tried quite a bit to figure out what is the most efficient way. We have also done our share on this, and in the end, I would confirm what you just said, right? You can go through the azide, which also gives you then access to a selenoester, but the conversion is not as clean and as efficient as with the pyrazole.
Okay, another group of questions is going is still sticking to selenium, and this goes more to the protecting groups of selenosystems. So what are the issues with the protecting group chemistry around seleniosystem in these ligation reactions? Can you comment a little bit on that?
Yeah, I mentioned that briefly along the way, that with selenocysteine, you have a lot of advantages in terms of reactivity, but also a couple of disadvantages based on this reactivity. One is the redox potential that leads to very quick formation of diselenides, which then gives you a peptide that is twice as big and sometimes can then also have, let's say, increased handling challenges due to that dimerization. And to prevent that, you typically carry through a protecting group. The conventional groups like the MOP group and so on that we find on seleноcysteine, they are typically removed by trans protection with thiopyridines and so on. And this works, but is again very much case dependent, peptide dependent. We have tried to find ways around this. So we now have explored much smaller thiols to form the thioselenium products with smaller protecting groups, also to increase solubility and so on. But overall, you need to be a little careful with that. Even then, if in the end you add reducing agent to have at least a certain percentage of free selenol in your reactions to do the initial attack on the seleno or sulfur or thioesters. While you do that typically with phosphines like TSAP, these can also lead to desalinization if you have too high concentration of these phosphines in there and so on. So these are all parameters in the reaction that you need to be aware of and play around with. And this is why I said that there are a couple of let's say, not really challenges, but I think a couple of parameters that you need to tightly control and to, at least in my opinion, control more tightly than the rather robust thiol chemistry, where you don't have to worry about that too much. If you control them well, the reaction is much faster at lower concentration, which is a big advantage.
Okay, I personally have a couple of more questions, but let's go on with that. One last to this topic is going from sulfur to selenium. Did you even do the further step going also to tellure? Did you ever have in mind playing with tellure? Just to mention that Professor Moroder already played around with that some 30, 40 years ago. And I remember him talking to me at some symposium about that. So I was eager to see whether you did something in this aspect.
No, we did nothing, and of course, it's kind of a logic extension of if sulfur is good, selenium is better. Why not go to tellurium? And see, the problem is toxicity, right? If you ask your students to work with tellurium, it's typically not something that they appreciate very much. And I'm also not sure if it would give you an additional benefit over the selenium, but the quick answer is no, we haven't tried based on these thoughts that I just mentioned, mainly safety, which should not be keeping us from doing it, because of course, as a chemist, you can handle such compounds as well. Yes. But the question is if there would be any additional benefit from what's in the literature so far. Not for ligation chemistry, of course, but for redox chemistry and so on before. At least the indications are not that great that it would help a lot.
Yeah. Okay, good. Here's another question. I think you mentioned increased chaperone activity for the heat shock protein 27 modified with archpyrimidine at the N-terminus. Do you notice any selectivity for client proteins, or is chaperone activity increased for all clients?
Definitely, that's an interesting question, because with these small heat shock proteins, they have a large variety of client proteins, but they seem to treat them and bind them. And in this case, there definitely is a difference. So we have previously typically looked at citrate synthase at one of the, let's say, poster candidates for aggregation and chaperone activity that have been used by many other labs, and where we had also direct access to assays developed in the group of Johannes Buchner. For this, we see an increased activity for these N-terminally modified protein versions. And then we extended this to a couple of other proteins that undergo what we would consider amorphous aggregation. So like the classical way proteins aggregate. And there we also see the benefit. We're not sure if this is really a physiologically relevant benefit, but in our in vitro assays, the improved chaperone activity, even to an extent where there is no aggregation. So I like to call them the super chaperones then, because they stop aggregation of certain client proteins completely. If they have, for example, 5 of these arc pyrimidine modifications in the N-terminus. And of course, this has something to do with a different binding mode and so on. But interestingly, if you go to different client proteins that have a different aggregation pathway, and we have tried that with tau, another protein that we like to work with. And of course, tau does not undergo amorphous aggregation, but forms fibrils during aggregation. And there we see no benefit, right? All the API modifications on the N-terminus have no impact. So it's as good or bad as a chaperone. And of course, we wanted to understand what's going on there. And there's a couple of hints in the literature that suggest different binding modi for these proteins. And also, we realized from looking at hydrophobic surfaces that are available in these modified HSP27 variants that has something to do with where the chaperone interacts with the client protein. So short answer, yes, there is a big dependence on what kind of client proteins. Right? And you cannot say this chaperone is a super chaperone for all proteins. It's again something where you need to consider the aggregation pathway in the interaction of the chaperone with this client protein.
Okay, thank you. There's one last, a bit more general question. How relevant are non-enzymatic protein modifications?
Yeah, that's a good question, and I try to address it briefly in the talk. There is a couple used as diagnostic markers, for example, in diabetes for long-term diagnostics, right? You look for non-enzymatic PTMs on heme proteins and so on. There is also indications that in aging, they play a role, like cataract and so on. There's also now new data from a couple of labs where they have looked at these kind of non-enzymatic modifications, for example, on histone proteins and so on, and have found indications that there are enzymes that recognize these modifications and maybe even be able to revert or reverse them, or at least change them into something that would make the protein functional again. And of course, this would be at least some very good evidence that they are physiologically relevant if you have the cell reacting to these modifications in a sense that they have derived enzymes or proteins that recognize them, can either remove these modifications or lead to a faster degradation of proteins that are modified in that way. And that would then tell us, okay, for the cell, some of these modifications are important because they need to recognize them, need to take action against these modifications. But this is something that is still under investigation. So we really don't know how many of them are relevant, if it's only for long-lived proteins where they are relevant. It's something that hopefully time will show, and not too much time, right? The next couple of years will hopefully help us when we have the appropriate probes in hand to actually go into cells and say, can we pull down proteins that bind these specific modifications? But of course, I also showed you there's a large class of these modifications. So we also need to make sure that we cover the ones that could be important and not miss major of these modifications. Yeah, but in general, I agree. The question is very valid, and we need to find more evidence that these modifications are really highly relevant.
And it's probably a topic for another talk or even series of talks. Yeah.
At some point, hopefully, yes.
Okay, so currently I do not see any further questions within the chat. Of course, any of people in the audience, if you have any further questions later on, you're always welcome to drop a line by us by email or give us a phone call. Just let us know. We will follow up anytime later. Christian, thanks very much again. It was a true pleasure that you shared your expertise and your view on this topic with us. I really liked it. And thank you very much for being our special speaker for this afternoon.
Thank you very much. I also enjoyed it. And thanks to everybody out there listening in and spending some time with us today. Thank you.
Yeah. All right. So with this, I would like to close our online workshop for today. As a follow-up, we will provide the recordings, which will be available on our website. And also thanks again for joining us. Thanks very much to all our speakers, Karen and Christian. And also a big thank you for the many people in the background who made it working so smoothly. Klaus Helm and his team. Thanks very much. We hope you had as much as fun as we had during this workshop. And we are looking forward to meeting you anytime in the future for our next one. Stay tuned. We will announce our next one on time on our homepage and on social media. So have a nice day and see you soon. Bye-bye.
Bye.
Bye-bye.